Okay, the last part of this topic A2 is on both brain death and how then things are measured, how we do brain. All right, start that again. All right, two. Okay, so this last screencast is on brain death or the pupillary reflex and also on methods of brain research. So if we take a look here, first here, this is actually the pupil reflex. I'm going to bring the notes up. All right, all up, sorry. The pupil reflex, again, is an involuntary reflex controlled by the brain stem, and it's under control by the autonomic nervous system we discussed earlier. What is the pupil reflex? Basically, it's the movement of the iris of the eye, which is the colored portion of the eye, which regulates the amount of light that reaches the retina. All right, in bright light, the parasympathetic nervous system takes over by the autonomic, which triggers muscles to contract and causes the pupils to get smaller. So larger iris, smaller pupil. Muscles, the eye that controls it, called circular muscles. In dim light, you have the opposites. Sympathetic nerves trigger muscles to contract and causes less iris, more pupil to show. All right, those are called radial muscles. So it's different contraction of a different set of muscles. All right, so if we go down and we talk brain death for a second, brain death is defined as the permanent absence of activity in both the brain stem and the cerebrum. Now, the brain stem is one little caveat there. The brain stem is responsible for all involuntary muscle responses in the body and may function alone to maintain homeostasis. So if you have a non-functioning cerebrum but a functioning brainstem, you may be kept alive by artificial, means and in a vegetative state. Brain death can be determined by medical professionals. Let's take a look here. There you can see the circular and radial muscles. Now notice the colored portion of the eye before we go on further with brain death. Colored portion is what gets larger or smaller. It looks like the pupil, but it's actually the iris. Circular muscles, radial muscles, two different kinds to contract. Let's test your consciousness, which I'd like to do every now and then in class. I'm not going to say to who, but you know who I'm talking about. Brain death can be determined by medical professionals by testing brain functions of specific autonomic responses. So the pupil reflex you see is on a 1, 2, 3, 4 response, eye opening. All right, so an autonomic that assesses brain, no response to pain, to speech, to spontaneous. All right, brain dead will not exhibit a pupil reflex, and they generally call that pupils blown. All right, you can see a verbal response, no response. You can see a motor response, reflex, the motor response. These are the three. Now, really here we're talking only about the pupillary reflex. All right, and so this here is actually called the Glasgow, Scotland, Glasgow Coma Scale. It determines whether a person basically is in what level of coma or is brain dead. All right, so that there is the pupil response. We take a look at a brain research as well. Pretty exciting here to take a look at brain research, and we go on. And there are three mains of brain research at this point. All right, an autopsy, which we would have been doing if we were doing animal experiments actually, but kind of an autopsy on it. So an autopsy is a postmortem. Okay, postmortem after death. An autopsy is something that's alive of a corpse via dissection in order to evaluate the cause of death. Comparisons can be made then between the brain, healthy and diseased tissue in corpses to identify affected brain areas. All right, a lesion now is an abnormal area of brain tissue which can be identified generally through an autopsy or via scans of the brain. You can a CT scan or an MRI can identify lesions too. The effects lesions can have can be difficult to identify as many functions can involve many brain areas, but the brain has capacity to relearn certain skills by rerouting. We talked about that earlier in the the section here by plasticity in the last section there. All right, so autopsy is one method of brain research. Another area of brain research, animal experiments, all right, can be used to identify function by stimulating organs with electrodes or removing the brain by lobotomy. All right, so you get a better understanding of brain disease here. So experimenting on animals involves less ethical restrictions than human studies, but it's still an ethical issue and standards do exist. Animal studies are limited by the difference of animals and human brains. So remember, we're talking about animal experiments versus human brain. All right. So animal experiments. Then lastly, we have your functional MRI. So that's an F functional magnetic resonance imaging, which records changes in blood flow within the brain to identify uh, active areas. And this is pretty cool because oxygenated hemoglobin responds differently to magnetic fields than deoxygenated hemoglobin. And so these differences in oxygenated hemoglobin can be represented visually, as you see in the second picture and the third picture here, to reflect differences in the level of brain activity. And this is a good thing because MRIs are not invasive and can be used to identify multiple brain regions involving complex and integrated brain activities. All right, it's used in such things as ADHD, dyslexia, as well as to monitor recoveries from stroke. All right, so you have three there. You have, again, the fMRI, autopsy, animal experiments.